In this video, we'll be looking at part 2 of macromolecules, which is going over proteins, or also we will call proteins polypeptides. I will use those two terms interchangeably. Proteins serve many functions in the body. I sometimes just like to say proteins do everything. Their shape determines their function almost every time. And so you'll see here this is a definite three-dimensional shape. And that shape is almost like you want to think of a tool, like a wrench or something like that, that has a particular job to do. Well, proteins are very similar. And their shape is the way it is because of how they are arranged chemically. Proteins are made of these molecules here. This is a monomer of proteins, and they are called amino acids. You've seen this before. You have your carboxyl group here, which, remember, this hydrogen wants to kind of break off and build up in solution, so this can be a carboxyl acid. And then over here you have your amino group, so you have your amino acid. Well, amino acids always look the same as far as this structure is concerned. The variable portion of the amino acid is called the R group. And this R group is what's going to cause a protein to have a structural difference from one other protein. The polymer of proteins is just called polypeptide or protein. There's not a special word for that, really. Here are some pictures of some amino acids. And the main idea here, you don't have to memorize these, that would be horrible. The main idea here is I want you to see all the R groups that are different. And they're the white boxes. So some R groups are just another hydrogen, like glycine. But some can get very complicated, like tryptophan here, the chemical in turkey that makes you go to sleep. And some have sulfur in them, which we've talked about, sulfur, proteins containing sulfur. And so sulfur is going to be a very important part of how proteins form and make their structure. Another amino acid that has sulfur in it is cysteine, another very important amino acid. So as you can see, those R groups can be many and varied. And so the only thing you need to know about them is that they exist. You don't need to have to memorize them or anything like that. I'll save that for your college biochem class. And so again, just to review here on the dehydration synthesis hydrolysis reactions. Dehydration synthesis, combination of two monomers, hydrolysis, breaking down two monomers. This happens with proteins as well. Combining two amino acids together, taking the water out, combine them together, and they form what's called a peptide bond. Hence, a polypeptide is multiple amino acids joined together a protein. A protein can have several dozen amino acids, or it can have several thousand. Proteins vary in size, and so we're going to look at how proteins are actually made. Proteins are structured in a four-part sequence. Some are just three, but a lot of them are four. And so that primary sequence is where you just have this string of amino acids. This is before the three-dimensional structure is made. I like to think of this as just a string of Mardi Gras beads. You can kind of see all the different amino acids here. This particular protein has 129 of them. And right now in this primary state, the protein hasn't formed a three-dimensional structure. It has just been made. And so the primary structure is just this simple string of amino acids. The important thing to note here is that the bond that's connecting all these are those peptide bonds, covalent bonds, very strong bonds. Now the secondary sequence is a little bit different. And so this is where those chains begin to bind together. And so this can happen in one of two ways. because of the chemical nature of those long chains, they are attracted to each other. They can do it this way, where they kind of form these coils, or it looks like a phone cord, if for those of you who remember what a phone cord is. And they kind of have this coil, and when they do this, this is called an alpha, Greek letter alpha, helix. All right, or they can form another type of structure where they kind of layer themselves like this, or fold over on themselves like this. And this is called a beta pleated sheet. And one string of amino acids could do both of these things, as you see with this structure here. You have some of these coils, you have some of these folded areas here, it could end the folded areas here, and so it could do any number of these things. The bond that causes these alpha helixes or beta pleated sheets are these attractions between these polar molecules. Remember, we talked about attractions between polar molecules. Those are called hydrogen bonds. And so these are not very strong bonds, but they're bonds that are easily connected to each other, like almost like a magnetic attraction. The tertiary structure of the protein is where some proteins stop. For some proteins, this is the final structure. What's going on with the tertiary structure is you have several of these secondary structures that are connecting together to form this kind of three-dimensional pattern. And they can be very complex, or they can be pretty simple. For some proteins, again, this is the final end. Now, tertiary structures can form many ways. As you can see, some of the bonds here, you have hydrogen bonds, you have these sort of interactions here, almost like a van der Waals interaction. You have these psionic interaction here, but the one that I want to point out the most as being the most significant are these two, this bond here between these two sulfurs. These are called disulfide bridges. And those disulfide bridges are strong covalent bonds. And this is what's going to help to stabilize the three-dimensional structure of the protein. When this structure breaks down, the protein itself breaks down. Again, that amino acid cysteine that I pointed out earlier is very important in forming these disulfide bridges. So again, some proteins are done at the tertiary structure, but for others, there is another structure called the quaternary structure. And the quaternary structure is where multiple tertiary structures combine together to make one single unit. And so the most common example that you'll see used of this is the hemoglobin molecule found in our own blood cells. Hemoglobin is its function is to carry oxygen. It does this with this embedded iron atom here. Each little section of hemoglobin has an embedded iron atom. And notice there are 4 subunits of that quaternary protein. So here you have multiple tertiary structures forming that single unit. And so here's another picture kind of showing you the protein here. You can see the disulfide bridges here in the yellow. The structures bound together. This is a different version of that. But it does show you the importance of those disulfide bridges, how everything works together to form this shape where that, I don't know what protein that is, but it's going to go and do something important. Now, that brings us to the concept of the denaturation of proteins. Proteins that have lost their structure are said to be denatured. There are several things that can cause a protein to denature. One of them is changes in pH, like a strong acid or base can cause a protein to denature. We're familiar with this because acids melt things and strong bases can melt things, and this melting is the denaturation of those proteins in the structure. Um, Temperature does this as well. Of course, the best example of this is when you fry an egg on a skillet. That egg, that albumin protein in the egg, that was that clear protein surrounding the yolk, now becomes solid and white. It has, the structure has changed, it's denatured, and it's kind of changed its chemical properties as well. And another thing that can cause proteins to denature is high salinity, like a high salt content. You think of like curing meats. They just cover them in salt and it cooks the meat, so to speak. And that cooking is done with salt alone. And so that is another example of denaturation. So one more thing about how proteins will fold up. There are molecules in the cytoplasm called chaperonins. And they're proteins themselves, but they are these little, I like to think of them as little floating changing rooms. Here's the polypeptide. This is the primary structure that is going to enter into this little capsule here. And it's going to get inside there and it's going to fold up. Now, what's the damage if the protein folds incorrectly? Well, the tool doesn't work, the function doesn't happen. So this is a very important structure. And so the chaperonin gives that polypeptide a safe area, an area that is suited for this sort of folding so that the protein can then fold correctly and then go and do its work. Now, since we're talking about proteins, it's appropriate for us to now talk about the nitrogen cycle, because nitrogen is featured prominently in proteins, being that all amino acids have that nitrogen in them, and proteins are made up of nitrogen, are those amino acids. And so we need to talk about how nitrogen is cycled in environments. All nitrogen comes ultimately from the atmosphere. Nitrogen is approximately 70% of our atmosphere. However, we cannot, nor can plants or any other organism, take in that nitrogen from the atmosphere and use it. We need help from, well, the only organism that can, and that is bacteria. And so as it rains, that nitrogen in the atmosphere will seep into the soil. And there are bacteria in the soil that are called nitrogen-fixing bacteria. Anytime you see this idea of fixing or fixation is what it's called, we'll see it later with carbon fixation. That is taking something that is not usable and making it usable by living things. And so nitrogen-fixing bacteria will take that bacteria and convert it into a protein or into a chemical that can be used. And that chemical is ammonium. And that ammonium is a protein that, or I keep saying protein, a chemical that can be used by other organisms. Ammonia can be absorbed by plants. Plants can use that to make DNA, can use that to make amino acids and proteins. But notice there are some organisms that really like that ammonium as well. These are called nitrifying bacteria. And through a process called nitrification, we'll turn that NH4, that ammonium, ultimately into NO3, which is called nitrate. And nitrate is another substance that plants can use. Plants can take those ammoniums, they can take those nitrates, they can make stuff out of them. And then there are bacteria that like that nitrate as well. These bacteria are called denitrifying bacteria. And through the process of denitrification, we'll turn that nitrate back into gaseous nitrogen, returning it to the atmosphere. And so this cycle is constantly happening in order to make nitrogen useful. Well, then you might think, well, what happens to dead peas and gophers when they die? What happens to the nitrogen in them? Well, they are decomposed by things like fungi and bacteria, and that decomposition turns the nitrogens into, or turns the nitrogen which is found in those bodies, which is usually ammonia, NH3, through the process of ammonification. turns that into ammonium, which can be then used by the plants and other things, and nitrifying bacteria and so forth. And so decomposition is another way of returning nitrogen back to the soil.